So thank you for this opportunity to present some of the work that we've been performing on bacterial epigenomics using long-read sequencing technologies. In particular, this is an example of the utilization of smart sequencing to evaluate the functional relevance of novel bacterial methyltransferases applied to a key human pathogen, Clostridioides difficile. So C. difficile is a spore-forming gram-positive anaerobic bacillus found ubiquitously in the environment and GI tracts of humans and animals. Its transmission occurs primarily via spores through the oral fecal route and can cause severe systemic infections, not only in immunocompromised individuals, but also elderly, antibiotic-exposed ones, and those in intensive care units. It is considered to be a common hospital-acquired infection with more than 250,000 infections per year, resulting in roughly 14,000 deaths, and more than $1 billion in healthcare costs per year. In this project, we performed the first comprehensive characterization of the DNA methylation landscape of C. difficile across a diverse collection of clinical isolates, with the purpose of better understanding the functional role of methyltransferases and corresponding methylation landscape in the sporulation process of C. difficile, which is the main route of transmission to human hosts, and eventually beyond sporulation. We used for this 36 clonal isolates from infected fecal samples obtained within the frame of an ongoing pathogen surveillance program at Mount Sinai Hospital. And our strategy involved, very briefly, a multi-scale analysis of methylome profiles, mutation complementation analysis, transcriptomic analysis, and comparative genomics and epigenomics. Through smart sequencing, we found a total of 17 unique high-quality methylation motifs across our genomes, the large majority of them being of 6MA type. And interestingly, one of these motifs, CA5, was consistently present across all of our isolates, as well as in the several hundreds of global C. difficile isolates that were found in GeneBank. Consistent with the presence of a highly conserved CA5 motif, we also identified a type 2 6MA solitary DNA methyltransferase present across our isolates, which was responsible for methylation at this motif, and which we called CAM-A. So here I show the effects of the delta CAM-A mutation in the mouse model. Mice were used to test each of the three genotypes, wild type, delta CAM-A, and delta CAM-AC, the complementation of delta CAM-A with the wild type mutant. And we found CFU levels to decrease steadily from day 2 post-inoculation to day 7. Notably, the delta CAM-A mutant showed CFU levels 10 to 100 times lower than those observed in the wild type and complement strains. And please note that this is a log scale. The bacteria were cleared from the feces roughly 6 days post-inoculation for the MTase mutant, while they were still detectable at day 6 and 7 for the wild type and complement strains. Taken together, these findings suggest that CA5 methylation events play an important role in C. difficile's ability to persist within the host intestinal tract. And this prompted us to perform a comprehensive methylome and transcriptome analysis of the wild type and MTase mutant strains. So C. difficile has an average number of around 7,700 CA5 methylation motifs per genome. To inquire if specific chromosomal regions are enriched or depleted for this motif, we used a multi-scale signal representation approach based on wavelet transformation. Briefly, in black, we have the distribution of CA5 across the leading and lagging strands of the genome, and in red and in blue, we have regions with a statistically significant over or under representation of motifs, respectively. We found CA5 motifs to be enriched in genes related to sporulation, which makes sense, but also membrane transport, transcription regulation, and cell wall proteins. To further characterize CA5 motif sites, we performed whole genome alignment with MOVE and classified each motif position in the alignment as either conserved or autologous, devoid of SNPs or indels, here shown in blue, variable orthologous, in which at least one genome contains a SNP or indel, here shown in green, and non-orthologous, shown in orange, typically arising by insertion of mobile genetic elements. We found the majority of motif positions, 75.4%, to be orthologous conserved. but still almost 14% of them were variable retardants. And these variable ones are interesting in the sense that they correspond to positions of methylation abrogation or on-off methylation switching mediated by genomic variation. Apart from on-off switching of DNA methylation mediated by genetic instability, the former can also arise through competitive binding between DNA methyltransferases and other DNA binding proteins, such as transcription factors. To test this, we built on the rich collection of C. difficile methylomes and perform a systematic detection and analysis of non-methylated CA5 sites. This is exactly what is shown here on this upper figure, the landscape of non-methylated motif sites across the C. difficile genome, which were found dispersed throughout the full length of it, yet with a minor percentage of positions given by these major peaks to be remaining non-methylated in at least 1/3 of the isolates, suggesting that competitive protein binding is expected to be more active in certain genomic regions. We next queried our genomes for all currently known transcription factor binding sites of C. difficile and found overlaps between prominent peaks of non-methylated CA5 positions and the TFBSs of COD-Y and ZIL-R. We have here the example of ZIL-R. And similar results were obtained when overlapping non-methylated CA5 positions and transcription start sites. Altogether, this shows that epigenetic regulation at CA5 sites is mediated both by small genetic expansions and contractions, as well as competition with regulatory proteins. To study the functional significance of methylation at CA5 sites, we used RNA-seq to compare the transcriptomes of wild-type C. difficile with that of the knockout mutant delta-Cam-A, both in liquid media in exponential and stationary growth stages, and in solid media, 9 and 10.5 hours post-sporulation induction. Of the almost 4,000 genes annotated in C. difficile, 1 to 9%, depending on the growth stage, were differentially expressed at 5% FDR, with effect sizes ranging between 13-fold for underexpressed and 80-fold for overexpressed genes. And the set of DE genes was enriched for the sporulation gene ontology GO term, which makes sense, as well as some additional terms, flagellum, ATP-coupled transport, UMP biosynthesis, which goes in line with previous CA5 distribution analysis. Finally, we attempted to explore the RNA-seq data further beyond the sporulation phenotype and the GO term analysis discussed above, with a special focus on biological processes critical to C. difficile infection. Specifically, we performed an overlap analysis between the list of D genes from our RNA-seq data, wild type versus delta-Cam-A mutant, 4 different time points, and those from published studies focusing on the colonization and infection by this pathogen. Shown here is one of those examples where we have our 4 data points shown in dark blue and those of another study in green, focusing on conditions that potentiate biofilm formation. The outer layer of the shell plot highlights the number of common genes between pairwise comparisons across the 2 studies, and its color is related to statistical significance. And indeed, we found multiple significant overlaps between our data sets, particularly at sporulation phases, with differentially expressed genes in conditions favoring the production of biofilm on a solid substrate. We tested this experimentally by crystal violet staining of growth supernatants of the 3 genotypes, and indeed, we found the MTase mutant to produce more biofilm compared to both wild-type and complemented strains. So the take-home message is that SMRT sequencing has been and continues to be a catalyst for the bacterial epigenomics field. I bring you here an example of the SMRT sequencing potential. Cam-A, which is a 6-MA solitary methyltransferase that impacts sporulation, biofilm formation, and colonization of C. difficile. And future work will delve into other bacterial persistent methyltransferases and methylomes to better understand not only their functional roles, but also their interplay with certain mobile genetic elements and effect on DNA structure. So the paper containing these and other results has recently been published on Nature Microbiology. Please go ahead and check it out. And I would like to finish by acknowledging the people from Fang Lab at Mount Sinai School of Medicine, at Tufts University, University of North Carolina, the Pathogen Surveillance Program at Mount Sinai School of Medicine, CNRS, Richard Roberts at New England Biolabs, and of course, the multiple sources of funding. Thank you very much for your attention, and I'm open to questions.